I started my education in a madrasa. Probably many of you know. I did my Dakhil and Alim, which is equivalent to SSC and SHC from Kumil Lalia Madrasa in 88 and 90. And then Dhaka University, biochemistry, BSc and MSc. And this is one of the pictures from the excursion. This is Shekhar sir and Imran sir and all of our friends. Hafiz is here today. Hafiz, I think I don't see you in the picture anyway. Then after my master's, actually I got a special viva and I left for my PhD. So when I almost finished 1 year of PhD, then I knew my master's results. So I was very lucky. So I did my PhD in Tokyo Institute of Technology. And this is Nagatsuta campus, Suzuka Dai. Onup is here. We are on the same building. And this is in my lab after my marriage in 2001. And many of you probably may know this is Yusuf, also a biochemist. So I brought him in my lab. And yeah, and this is one picture in a Sakura party in the campus, Okayama campus in Tokyo. So Onup is here, biochemist. And this is Azhar, also a biochemist. And then after my did PhD, I got a JSPS postdoctoral fellowship for 2 years. And then I moved to Europe, a university called Joseph Fourier University. So I did 1 year of postdoc there and studying peptide-mimetic drug design. And then 2005, this is the first day I came to Melbourne in my current institute. So 2005, I was like this. So this is a brief journey from 90 to 2000, till today. So this is where is Florey Institute of Neuroscience and Mental Health in Parkville campus in Melbourne. And this is my lab. I would like to acknowledge the contribution of all the lab members. Most of the work I will present today is from Dr. Liu and Praveen. And some previous students, Doris and Patil, I will talk some work from them as well. And I would like to also acknowledge the contribution of my collaborators. So Ross Lab is specialized in cell signaling, particularly GPCR cell signaling. And John Farnes group is expert in gut physiology. And Krishan Samuel's lab is on matrix biology, mainly fibrosis physiology. And Brian Forbes' lab is specialized in insulin pharmacology, and Paul Guri's lab for structural biology. And I also do some work with John Wade and Professor Kajihara. He's from Japan, mainly his expertise in carbohydrate chemistry. And my lab and work is also funded by Novartis, Takeda, and 2 biotech companies, Peritech and Enzytech, and also Australian government organizations like NSMRC and ARC. So there are 10 members of insulin-like peptides. So we all know about insulin is for the treatment of diabetes. So when I talk about insulin and insulin-like peptide, people get confused. They think all peptides are for diabetes. Actually, this is not the case, because insulin is for diabetes and all other members, they have different receptors and therefore different function. All these peptides have 2-chain 3-disulfide bond, except IGF-1 and 2, they are linear peptide, but they have 3-disulfide bonds as well. But if you look at the receptors wise, these 3 peptides, they interact with the tyrosine kinase receptor, whereas these 7 members we call relaxin family peptides or insulin-like peptides. They signal through G protein coupled receptors. So this Java said earlier that these are very important target. And currently, all the drugs you see in the market, more than 30%, almost 30% of drugs actually target these receptors. So these family of peptides are very important therapeutically. There are some challenges of this insulin-like peptide drug discovery because they are 2-chain 3-disulfide bonded peptide, and it's very difficult to produce in large production. Some peptide you can make recombinantly, but some of the peptides, for example, insulin 5, very difficult to recombinantly produce. And also, these peptides have a very short half-life. Their pharmacokinetic properties are poor. And therefore, if you want to modify to improve the pharmacokinetic and thermodynamic properties, it's very difficult. And it becomes expensive as a drug. And some of the peptides also have cross-reactivity with other receptors. So therefore, my team is much more interested in designing a small peptide which will mimic the function of these big molecules. So we call them simplified modified peptide or peptidomimetics. So can we make a peptide which is very simple, but will mimic the function of this 2-chain 3-disulfide bonded peptide? So peptidomimetics are becoming very popular. They fall into between these two group of drugs called small molecule and biologics. And small molecules, they are orally available, cheap, but they can be toxic because they can have off-target effects. On the other hand, the biologics are very target specific and therefore less toxic, but they have to be injected and often very expensive. But peptidomimetics, which falls between this category and molecular size is in like 500 to 5,000, we call them peptidomimetics. They are kind of modified peptide. They can be injectable, but also it is possible that you can modify this peptide to be orally available. And they can be highly specific and therefore less toxic and less expensive. And current FDA approval rate for this kind of peptide is twice than small molecule. In some cases, when protein-protein interactions is concerned, it is very difficult to target protein-protein interaction by small molecule. For example, if you see here, these two protein and they bind, they interact here at the surface, they mediated by alpha helical interface, you see here. But this pocket, this binding pocket is very difficult to target by a small molecule because small molecule cannot contact all the binding pockets. So we call this kind of target as an undruggable target. So now, say for example, if you want to make this peptide as a drug, what happens is you take this out of this pocket and it becomes unstructured because this structure is conferred by the whole protein. So this area of the protein is important to have this conformation. So when you take out of context, it becomes unstructured. But as a chemist, you can make this unstructured peptide structured by stapling. It's like you have two linkers and make a staple here. And now this helical peptide, you can see here, it does have all the contacts. So this small peptide can target these binding pockets. That's why this kind of analog or this kind of peptide mimetics are becoming very popular nowadays. So to make peptide mimetics of insulin and insulin-like peptide, we need to understand the structure-activity relationship between the ligand and receptor. Insulin-like peptide, you can see they have extraordinary structural similarity. The B chain of this family of peptides is overlapping, you see, except the termini. The same for the A chain. And from the SAR studies, we know that all this family of peptide actually, when they interact with the receptor, they use the B chain. So most of the cases, A chain is working like as a scaffold to give the support for the B chain to have this conformation. And this is human relaxin 3, a neuropeptide. From SAR study or a structure function study, what we have seen here, you can see this is molecular modeling. This is the B chain of human relaxin 3. And A chain is on the top. And all the binding residues actually at the interface of this helical B chain. So you see there is no A chain specific residues have any contact with the receptor. So if we can make a helical peptide like this one, it is possible that we can make an agonist or antagonist just based on this B chain. So we have designed this peptide. This is derived from the B chain of relaxin 3. And we introduce two unnatural amino acids here called S-pentenyl alanine. And then a reaction called ring closing metathesis, we staple the two amino acids. And this peptide become very structured, helical. And it did mimic the function of H3 relaxin. So if I can show here is like, without stapling, you see the peptide is unstructured. When you do ring closing metathesis, the peptide becomes structured. One limitation of this kind of reaction is, many of you will know that when there is a double bond, you will have a cis-trans geometry. So two isomer here. And because you make the peptide on resin and then clip and you have this modification, the yield is often very low. And you have to use a catalyst that is very often difficult to remove. So my team is trying to develop a high yielding stapling method. And very recently, we were able to make this peptide, but this stapling method I am not disclosing because of some patent issues. But you can see the activity of this peptide. So let's go back to this peptide. This is called hydrocarbon staple, and we call it peptide 5. So I'm just comparing my new compound with peptide 5. So this is peptide by cyclic AMP. This is how to say in RxCP3 receptor assay. So peptide 5 have this activity if you compare with control peptide. So control peptide will have just linear sequence without any stapling. Now look at my new analog, which is non-covalently stapled, but it is stapled. Is the activity is as good as peptide 5. And look at the yield of this peptide. When human relaxin 3 has 2% or 5.7% yield, this single chain peptide has almost 50% yield. And the peptide has no disulfide bond, single chain, only 18 residues. This is agonist. And we also have developed antagonist, which is only 14 residues. And look at the stability of this single chain peptide. If you look at the control peptide, is very quickly degraded. But this staple peptide, the half-life is almost like over 350 minutes compared to 20 minutes. So the stability of the peptide also increased. The activity is similar to peptide 5 or relaxin 3. So this kind of compound, agonist or antagonist, they will be very valuable tool or drug lead for the treatment of various diseases. For example, we have shown before in Florey that relaxin 3 can treat anxiety and depression in mice, and the antagonist peptide can treat alcohol addiction or drug addiction. So these are neuropeptide, target is in the brain. It's very difficult to target peripherally, so the people use direct injection into the mice. But because we have now small peptide, it is very easy to modify so that it can cross the blood-brain barrier. So lots of opportunity to work with this peptide in the future. So I want to talk some about relaxin 2. This is another peptide. It is similar to human relaxin 3, but the receptor is different. The receptor for human relaxin 3 was relaxin 3 receptor RXAP3, and this receptor is called RXAP1. And from SAR study, it shows that both chains have some interaction with the receptor. Unlike relaxin 3, that has only the B chain was binding to the receptor and activating the receptor through the B chain. But for relaxin 2, from SAR study, it looks like B chain and A chain both have some interactions. But B chain, mainly the B chain is the driving force that has the primary binding residues. So can we develop B chain only RXFP1 agonist or antagonist? So in 2005, this group has tried to make constrained peptide, staple peptide by disulfide bond. And some of the peptide you can see have helical structure, but none of the peptides are active. So we recently revisited these studies, and what we have seen that B chain and all the analogues are very insoluble. So if I want to show you some solubility issue. So this is the B chain of human relaxin 2. It has 29 residues, net charge 0. Look at the solubility. So it is possible that when they put the peptide in the assay, the peptide crash out of the solution. So I tried to make it a soluble form. So what I did, so I used 4 residue at the C terminus. Where it comes from the C peptide. Here there are 4 residues that is cleaved here. So I put back those 4 residues. On the other hand, in addition, I cleaved these 6 residues from this terminus. Now the net charge of this peptide becomes 5 and the peptide is soluble. And it is also a native peptide. There is no like unnatural or non-natural amino acids or something not part of the human relaxin. So this peptide was very soluble. And there are lots of data I am not showing because I wanted to tell you a full story of my work, a little bit of from here and there. So we tested this peptide in cell-based assay, and we found this peptide is very active in natively expressing RXFP1 cell. And we tested this peptide in 4 disease models like 2 heart fibrosis, lung fibrosis, and renal fibrosis, 2 animal models, and by a different route of administration and different time period. And you can see in all cases, B7-33 either prevented or reversed fibrosis and improved organ functions with similar efficacy to human relaxin. And in most cases, it was better than ACE inhibitor, which is a drug for heart failure. This is very interesting. Probably some of or many of you may know this biased agonism is very popular in drug discovery nowadays. So what biased agonism means? The same cell, the same ligand can have different signaling profile. If you look here, this is Hubeck cells, and B7-33 and H2-relaxin both activate the receptor cyclic GMP signaling, but look at the cyclic AMP signaling. B7-33 does not have any activity. So sometimes bias could be a good thing or sometimes bias could be a bad thing. So it looks like B7-33, this cyclic AMP activity is not necessary for people with fibrosis or heart failure. I'm going to give you some examples. For example, we saw that B7-33, if you compare with H2-relaxin, it has a strong antifibrotic effect, vasodilative effect, anti-inflammatory, anti-hypertrophic effects. But they're similar. It's not very interesting. You want to see some differences because it is a bias. We think that because it does not activate the cyclic AMP, it does not promote tumor growth. So human relaxin was shown many literature that if you activate human relaxin, you see tumor growth effect, but 33 does not. And also B7-33 does not react with or cross-react with another receptor called RXCP2. So this is RXCP1 specific, cheaper and safer, and very small scaffold to modify to improve their PK properties, and therefore it could be an ideal template for further development. And what is the therapeutic potential of this peptide or this kind of mimetics? So fibrosis has actually no drug. If people develop fibrosis in organs like heart or kidney, actually you can have some drug which can delay the progression, but ultimately there is no drug. So B7-33 could have the potential in the future, probably, if we can make it as a, how to say, effective therapeutic. It can be a drug for antifibrotic, for drug as an antifibrotic therapy, also could be useful for heart failure. This is another application of this peptide, could be when people use medical device, like implantable medical device. So there is foreign antibody response. There is fibrotic encapsulation around the device. So recently, my team collaborated with CSIRO Australia. They are expert in device development. They use this compound, B7-33, and coated the surface with this and a polymer to slow release of B7-33 upon time. And they have shown that B7-33 reduce fibrotic encapsulation by 50% after 6 weeks. So this is quite potential application for implantable medical device, for example, surgical mesh or glucose monitor, blood glucose monitor or something like that. So we are working with this CSIRO, this group, to develop medical antifibrotic medical device. This is one of the peptide I was always very interested. It's called insulin-5, and the receptor is called RXCP4. This is a gut hormone. This is quite interesting that the hormone and the receptor is in the colorectum. And previous studies, my group and other people showed that it has some effect in insulin secretion and also in appetite regulation. However, the effects were modest, and in some animal models, the results was not reproducible, like different animals showing different effects. So recently, my group collaborated with the other group at Melbourne University, who are gut physiology expert. So we think because of the anatomical location of this insulin-5, it's in distal colon, colonic L cells, and it has a role in secretion of water and nutrient. And we think that probably insulin-5 is a colon motility regulator. Well, the problem with this peptide is, as you can see, the yield, the production yield is like 1%. It's almost impossible to produce enough amount of peptide to do preclinical studies. So again, doing a lot of SAR study to minimize the structure of insulin-5, where we could show this peptide has 2 disulfide bond instead of 3 disulfide human insulin-5. The binding affinity is similar, it's much more simple. Yield is 14% compared to 0.8%, like 17.5-fold higher. So this gives us an opportunity to validate the target because now we can produce enough RXCP4 agonist peptide to validate the target in animals. So last year with John Farney's group, we try to see if we can prove our hypothesis. So what we did was a lot of cell signaling studies. This compound was active, so we put it in the animal. We did some bead expulsion experiments. So what is bead expulsion experiment? We put 3 millimeter round bead inserted at the distal end of rectum for 2 centimeter. So it goes there, then take out this and measure the time, how long it takes to take out the bead. So if there is a propulsion, the bead will come faster. If there is colon motility, if it is contracting, the bead will come faster. So you can see here this agonist peptide, which I have shown before, with just 2 disulfide bond, but still 2-chain peptide, dose dependently reduce the bead expulsion time. So first, we injected the mice with loperamide to make it constipated. You can see the bead expulsion time is like 1000 seconds, right? So you start injecting with the agonist peptide, and you can see bead is coming faster. The more peptide you are injecting, bead is coming faster. That means there is a propulsion there, colonic propulsion. There is contraction there. So bead is coming faster. That means your agonist is working, reversing the constipation. So loperamide is an opioid agonist. You increase the bead expulsion time, and your agonist is reducing the time. So that means agonist has the capacity to cure constipation. You can say something like that. But when the animals doesn't have the receptor RXCP4, you can see there is no effect. So that means the effect you see here is RXCP4 specific, the receptor specific, because the knockout mice does not show this effect. Up until now, we did not have any antagonist in our hand or anyone in the literature in the world. So my team is last 10 years was trying to develop an antagonist for this receptor. And this is the first time this year we reported the first antagonist of this RXCP4. So what we did was, again, a lot of SAR study behind. We modified this one residues at the C terminus of the B chain. So if you remember the last slide or slide before, the agonist peptide has 2 chain we call insulin 5A13. It has 2 disulfide bond. So we modified this peptide from agonist to antagonist. So these residues arginine we modified with some arginine mimetic. Only difference is the length of the side chain if you look at them. So this is homoarginine, arginine, norarginine, and so carbon chain is just different. So this, hello? Okay. I can see some vibration.
We can hear you all right. Akhter, we can hear you.
Okay. Because I can see a lot of vibration coming or echo coming anyway. So if you look at them, I do not know why I hear this. Can you hear me now?
Yes, we can.
We can hear you.
Okay, thank you. So if you look at this residue, as I said, this one residues. So these residues are very important for binding and activation. This is as I said to you, I am not showing you loads of data because it's impossible to show you. So lots of SAR study behind. So we found this residue so important. If we modify this, it becomes inactive. So what we thought was like to mimic the side chain, but change the length of the amino acids. So we modified this, and one of the peptide turned out to be an antagonist, a very good antagonist from cell-based assay. The first antagonist of this receptor ever reported. So you can see here, this we tested in males and females. So these are combined results. Again, bead expulsion experiment, loperamide increase the expulsion time. So you can say this is a constipated. And then we treat with agonist, you reverse the action of this loperamide. That means you cure constipation, you can say. But when you inject with both agonist and antagonist, you see you reverse the action of agonist. So this antagonist peptide, which we developed, can block the effect of this antagonist peptide blocks the effect of agonist. Then we wanted to see, we have a hypothesis that diarrhea is most of the diarrheas are due to bacterial metabolite. That is overgrowth of bacteria in the colon. So, and those bacteria metabolites are short-chain fatty acids. So we introduced short-chain fatty acids to the mice, and you see it has less expulsion time. But when we added antagonist and short-chain fatty acids together, you can see that you can block or reverse the bacterial metabolite-induced diarrhea or accelerated propulsion. So we can say that our antagonist is able to block the action of short-chain fatty acid. Now, these, as I said earlier, those peptides have two-chain, 2-disulfide bond. The yield is higher, like 17.5-fold higher than native insulin 5, but still they are challenging because two-chain, 2-disulfide bond are not very easy. Can we make single-chain peptide by using the stapling chemistry we developed in my lab? Yes. We just recently we are able to make this peptide. Again, I'm not showing the structure, what kind of stapling it is. And this single chain peptide is about 100 nanomolar potency, but native peptide is around 10 nanomolar. So it's still not that effective, but 100 nanomolar is a very good potency. Look at the yield. So where native insulin 5, you can produce 0.8% yield, and this yield is 40%. So because it is a single chain, right? And look at the, as I said, 100 nanomolar potency. So we are excited to see what is its effect in bead expulsion tests. Just this data came out 2 weeks ago. Very exciting that we can see this is the two-chain agonist peptide. You see the efficacy. It does reduce the bead expulsion time compared to, you see, loperamide. So 1000 second, but agonist two-chain peptide reduces the time. We have seen this before, but our single-chain agonist, which is very small, has similar effect. But of course, the dose is higher because we know the potency is low. It doesn't matter because we can make kilograms of this peptide now. So we can do even human trial if we want, if it works in the future. So these are very good tools for us to not to dissect what this peptide is actually doing in the colon. We have access to now both agonist and antagonist, and we can really accelerate this program. So do we need drug for this this kind of disease? Actually, people do not know. Constipation is very underappreciated disease. People don't talk about it, but it is not a very good disease. Laxative, most of cases do not work, and people don't talk about it. Sometimes people has to go to hospital. Just 2011, there is a report from US that every year in US, $1.2 billion is spent in the hospital. People visit just to get out of their stool. So this is very serious disease. And the best drug in the market, which is called prochloropride, is just like 20% better than placebo. Actually, it's not effective at all. It is safe, but not very effective. So there is a need for new drug for constipation. And likewise, diarrhea, some kind of diarrhea like IBS and IBD, diarrhea, there is actually not good treatment nowadays. So normal diarrhea, yes, but when diarrhea is due to IBS or IBD, there is no good treatment, or the treatment they have is called allosterone, is severe side effect. So this kind of molecule may have a future. We do not know. It's very early stage. So we are excited, but we have to go very far. This is my final slides of insulin. This is one of the molecules I am always very interested in. 100 years of insulin discovery, still there is no small molecule. We still have to inject every day. There is no peptide-mimetic. The only reason is there are 50 amino acids in this compound, almost like 45 of the amino acids somehow interacting with the receptor. So it's so difficult that you can make or you can use a small molecule. So we have to live with 2-chain peptide with 3-disulfide bond. It could be long lasting, fast acting. But are these molecule ideal? No, because all this insulin have a short shelf life. We have to put in the fridge because they form fibrils, and temperature also accelerate the fibril formation. And can we make a non-fibrillating insulin? We just recently published this paper last year, and we showed that yes, we chemically can produce insulin which can be non-fibrillating. So I'm not going to talk details about the chemistry. So what we did was we made a new insulin scaffold we call thiol insulin. Instead of 6 cysteine, we have 7 cysteine in this molecule. Interestingly, I incorporated this cysteine on purpose because after 3-disulfide bond formation, this free thiol, you can conjugate anything you want, like fatty acid, sugar, or albumin, whatever. So this opened up a new area of insulin chemistry. And interestingly, I never expected this molecule will be stable because when you have a free thiol, usually it does clip the disulfide bonds. But we found this peptide is extremely stable. It does not reduce the disulfide bond. What it does is like it dimerizes. If you put in a high concentration, another insulin molecule make a disulfide and becomes a dimer. That's why you can see whatever condition we use to conjugate this sugar, we always get this insulin dimer. It's like that's why I will talk about this little bit later. I wanted you to show that when we conjugated with this sugar molecule, we always get this dimer, like 30% yield. So anyway, so why sugar? Sugar is very high, it has very high solubility. So it makes the insulin molecule very soluble, and it has hydration effects. And we hypothesize that such a molecule will have less fibril formation tendency. So first we characterize in insulin receptor, does we alter the receptor affinity? It looks like affinity is very strong. So you see some reduction in the affinity, but the affinity is still at 1 nanomolar. So it's very, very potent. And glucose lowering effect by IP injection or subcutaneous, they have this glycoinsulin molecule has a similar glucose lowering effect like insulin or glargine. And the structure, if you look at the secondary structure of glycoinsulin and insulin, they have similar secondary structure. And serum stability, insulin is degraded, you can see 60-70 minutes, hours, insulin is degraded. And it's in vitro. In vivo is quite different because you have an issue of clearance, like kidney clearance, liver metabolism, lot of PK things going on. But this is just in vitro against enzyme. But in vitro, when you see glycoinsulin is more stable than insulin. This is what it is interesting here. We use different concentration of insulin, 50 micromolar and 200 micromolar. And the temperature is normal temperature and 60 degree. These are data from 60 degree. You can see that insulin at low or high concentration, they form fibril. You can see visually. So this is called atomic force microscopy. You can see the fibril formation immediately. So, but look at the glycoinsulin. We see 6 hours, 8 hours, there is no fibril formation. So these were the data we published last year, but we were curious if this is the case, we can use this glycosylation chemistry for other drug. For example, glucagon. Glucagon is also very known to have fibril. They are so severely fibril forming that currently used drug is not in solution. So it's always in the lyophilized as a powder, because if you make a solution, it becomes fibril, and after some time, it is no more useful because it aggregates, it couldn't be toxic, it has no activity. So we wanted to test this peptide. So we chemically modified this peptide and we choose these residues where we think it will not alter the activity. And again, there are a lot of SR study behind. So we made this glycoglucagon, and you can see the activity is similar to glucagon. And look at the fibril formation study, 1 day, 2 day, 3 day. Even though you can see some on the glycoglucagon, but look at the extent of they are less compared to glucagon. Glycoglucagon is less prone to fibril formation. So what is this therapeutic application? One application could be, they could be used in insulin pump. Because the pump, when people use insulin, they block the catheter, the pump, and that is dangerous. You can have less dose. So this kind of molecule can have application in insulin pump. Glycoglucagon, as I said earlier, currently they are lifeless powder, and you can have now liquid formulation, this kind of glycoglucagon molecule. And more importantly, scientists are trying to develop bihormonal closed loop. For example, one device, you will have both insulin and glucagon. And whenever you have so much sugar in your body, this device will deliver insulin. And when you have hypoglycemia, the device can deliver glucagon. And this kind of development is, how to say, not in the market. We think that a soluble form of insulin, non-fibrillating and non-insulin and glycoglucagon will be very useful for the device, because then you do not block the device with non-fibrillating insulin and glycoinsulin. So this could be potential application in the future. And as I said to you earlier, this is quite interesting. This molecule, I was quite amazed to see that why it forms so quick dimer for dimer. And dimer is like, it's a big molecular weight. It's like more than 12,000. So if you have a big molecular weight molecule, the kidney clearance becomes slow. So it could be long-acting molecule. So I was interested to see, to study and characterize this molecule. So this is my hypothesis. Because it is quite quickly forming insulin, thioinsulin is forming dimer. So I think we can make high-yielding insulin dimer. And it could be biologically active. To date, in the literature, there is no dimer insulin reported. Two papers reported previously, they both inactive. Because their position of dimerization was different than this one. And this is, you can call serendipity, because it was not what I designed. It is something I found and I wanted to investigate. And this result just came out few weeks ago, 2 weeks ago, I think. Look at the data, quite interesting. This is thioinsulin monomer, and this is 5 minutes. Just we did in water. You have this thioinsulin monomer, and upon time, look at monomer is becoming dimer. 6 hours, 8 hours, 10 hours. We do nothing, no reagent, nothing, just in air oxidation. You put in the Eppendorf tube and just monitor by SPLC. And after 18 hours, there is no monomer left. This peak you see is purely dimer. And I didn't bother to purify this molecule because it's like 98%, over 98% pure from insulin monomer to dimer. So then we did this characterization in insulin receptor assay. Look at the activity. It's as active at dimer is as active as insulin molecule. And this is quite interesting as well. Here, this is called IGF-1. So IGF-1 has a long history of cancer promoting effect. So this insulin also bind to IGF-1 receptor. But our molecule, which is insulin dimer, compared to insulin, it has, if you look at this red one, this is called glargine. Glargine is one of the blockbuster insulin drug. So compared to glargine, dimer has less binding affinity, 2.5 fold less. So we think it could be potentially little safer than glargine. But having said that, I have to be cautious. Glargine is not reported to be not safe. Millions of people are using, but there are some, occasionally some paper comes out, oh, glargine have this problem, but really it is not a big issue. But good thing is, you see, dimer has less affinity for IGF-1. And in insulin tolerance test compared to glargine, you can see that our molecule is also active. Initially, it is slow acting, but if time goes, if you compare with glargine, you can see the effect, how to say, sustained releasing effect. So it is not a good asset to say it is long acting, but regardless, it is it is biologically active, you can say. And if we look at the two-dimensional structure, which is helical structure, our insulin dimer is little bit less structured, but it does not affect the binding potency, as I have shown you earlier. In serum stability studies, insulin dimer clearly at these 3 points, you can see 40 hours, 60 hours, 100 hours, this dimer is more stable in rat serum. So on this note, this is the last slide, I believe. This is the first report on biologically active dimer we reported. And it's a very high yielding, unprecedented high yielding of any insulin analog. Imagine we can make insulin monomer by recombinant method. And you just put in water and it will form dimer. You don't need to even purify. All of the assay I have shown here, animal testing, cell-based assay, all were done crude dimer. We did not purify them. So this peptide synthesis and structure-based rational drug designs, I wanted to tell you that these are indispensable tools for peptide and protein research. They enable structure function study to be determined for peptide ligands. And novel peptide-mimetics like agonist and antagonist with superior activity like serum stable, thermostable, non-fibrillating can be designed and developed. These peptide-mimetics are important research tools and potentially drug leads. Thank you. So I'll be happy to answer if you have some questions.